Welcome back to another Sleepy Steve story. Before we drift off tonight, remember to subscribe to help Steve-O out, and I will keep making more sleepy stories to help you study gently, revise quietly, and settle your mind at the end of the day. Tonight, we are continuing our slow journey through VCE biology, and we are staying inside Unit 3, Area of Study 2, where cells transform energy and regulate the chemical reactions that keep life moving. In the last 2 episodes, we walked through photosynthesis and cellular respiration. We watched light energy become chemical energy in the chloroplast, and we followed glucose as it was broken down in the mitochondrion to release energy for ATP. Tonight, we slow down and look at the helpers that make those pathways possible. We are looking at enzymes and the way biochemical pathways are controlled. This is one of those biology topics that seems simple at first. Students often remember the phrase enzyme equals biological catalyst, and that is a good starting point. But for VCE biology, we need to go further. We need to understand how enzymes work, why their shape matters, how they lower activation energy, how conditions affect them, and how inhibitors and helper molecules can change the rate of a reaction. So, before we drift too far, let us set our coordinates. Tonight's episode covers enzyme structure and function, active sites, substrates, products, enzyme-substrate complexes, activation energy, denaturation, temperature, pH, competitive inhibition, non-competitive inhibition, cofactors, coenzymes, and the regulation of biochemical pathways. That sounds like a lot, but we will take it gently, one small reaction at a time. Start with the big idea. A biochemical reaction is a chemical reaction that occurs inside a living organism. Cells are constantly carrying out biochemical reactions. They build molecules, they break molecules down, they rearrange atoms, they transfer energy, they join amino acids into proteins, they split glucose during respiration, they use carbon dioxide during photosynthesis. The cell is always busy, but it does not do this randomly. For a reaction to occur, the reactants need enough energy to begin. This beginning energy is called activation energy. You can think of activation energy as the small hill a reaction must climb before it can move forward. Even if a reaction is useful and possible, it may not happen quickly enough at normal body temperature unless something helps it begin. That is where enzymes come in. An enzyme is a biological catalyst. A catalyst increases the rate of a chemical reaction without being consumed by the reaction. This means the enzyme helps the reaction happen faster, but the enzyme itself is not used up. After the reaction, the enzyme can be used again. Most enzymes are proteins, because proteins fold into specific three-dimensional shapes. Enzymes have very specific structures. Their shape is not just decoration. Their shape is their function. If an enzyme's shape changes too much, its function can be reduced or lost. That is why the phrase structure determines function is so important in biology. Every enzyme has an active site. The active site is the specific region of the enzyme where the substrate binds. The substrate is the reactant molecule or molecules that the enzyme acts upon. When the substrate binds to the active site, an enzyme-substrate complex forms. The enzyme then helps convert the substrate into product or products, and those products are released. A classic way to remember this is enzyme plus substrate forms, enzyme-substrate complex, then enzyme plus product. The enzyme is present at the start, present during the reaction, and present at the end. It helps the reaction occur, but it is not permanently changed by the reaction. The active site is specific. Not every substrate can bind to every enzyme. The shape and chemical properties of the active site must match the substrate closely enough for binding to occur. This is sometimes explained using the lock and key model, where the substrate fits into the active site like a key fitting into a lock. A more flexible explanation is the induced fit model, where the active site changes shape slightly as the substrate binds, allowing the enzyme and substrate to fit more closely together. For VCE biology, the important point is that enzyme action depends on the complementary relationship between the enzyme's active site and its substrate. If the substrate cannot bind properly, the reaction will not be catalyzed efficiently. Now, let us return to activation energy. Enzymes lower the activation energy required for a reaction. They do not change the overall energy released or absorbed by the reaction. They do not make an impossible reaction possible. Instead, they provide an alternative reaction pathway with lower activation energy, so the reaction can occur more quickly under the conditions inside a cell. This matters because living organisms cannot simply heat their cells to make every reaction faster. Too much heat damages proteins and membranes. Instead, organisms use enzymes to speed up reactions at temperatures that are compatible with life. Enzymes allow life to be chemically active without becoming chemically chaotic. In photosynthesis, enzymes help control reactions in the light-independent stage. In cellular respiration, enzymes help control glycolysis, the Krebs cycle, and the electron transport chain. In DNA replication, enzymes help unwind DNA, add nucleotides, and join fragments. Across biology, enzymes are everywhere, quietly making controlled life possible. Because enzymes are proteins with specific shapes, they are affected by their environment. Temperature is one important factor. At low temperatures, molecules move more slowly. Substrates and enzymes collide less often, so the reaction rate is usually lower. As temperature increases, molecules move faster, collisions become more frequent, and reaction rate usually increases. But this only happens up to an optimum temperature. The optimum temperature is the temperature at which the enzyme works most effectively. Above this temperature, the enzyme may begin to denature. Denaturation means the three-dimensional shape of the enzyme changes. If the active site changes shape, the substrate may no longer bind effectively, and the rate of reaction decreases. Denaturation is not simply the enzyme getting tired, it is a structural change. The bonds and interactions holding the enzyme's shape are disrupted. Once the active site loses its specific shape, the enzyme may no longer catalyze the reaction. In exam language, increased temperature beyond the optimum can cause denaturation, altering the shape of the active site and reducing enzyme-substrate complex formation. pH also affects enzymes. Each enzyme has an optimum pH. Some enzymes work best in acidic conditions, others in neutral or alkaline conditions. If the pH moves too far from the optimum, the enzyme structure may be affected, again altering the active site and reducing the reaction rate. This is why enzymes in the stomach and enzymes in the small intestine can have very different optimal conditions. Substrate concentration also matters. If there are more substrate molecules present, the chance of enzyme-substrate collisions increases, and the reaction rate can increase. But this does not continue forever. Eventually, the enzyme molecules become saturated. That means most active sites are occupied most of the time. Once all available enzymes are working as fast as they can, adding more substrate will not increase the rate unless more enzyme is added. Enzyme concentration can also affect the reaction rate. If there is plenty of substrate available, adding more enzyme provides more active sites, and the reaction rate can increase. But again, there are limits. If substrate becomes limiting, adding more enzyme will not continue to increase the reaction rate. So enzyme activity depends on conditions. Temperature, pH, substrate concentration, and enzyme concentration can all influence the rate of an enzyme-catalyzed reaction. In biology questions, the safest approach is to link the condition to collisions, enzyme-substrate complex formation, active site shape, and reaction rate. Now let us consider inhibitors. An inhibitor is a molecule that reduces or prevents enzyme activity. Inhibitors are important because cells do not always want reactions to happen as quickly as possible. Sometimes a cell needs to slow a pathway down. Sometimes a substance blocks an enzyme accidentally. Sometimes a drug works by inhibiting an enzyme. Inhibition is a key part of regulation. Competitive inhibition occurs when an inhibitor competes with the substrate for the active site. The competitive inhibitor has a similar enough shape to the substrate that it can bind to the active site, but it does not react in the normal way. Because the inhibitor occupies the active site, the substrate cannot bind, and fewer enzyme-substrate complexes form. The word competitive is important. The inhibitor and the substrate are competing for the same place. If substrate concentration increases enough, the substrate may outcompete the inhibitor. That means the effect of a competitive inhibitor can sometimes be reduced by increasing substrate concentration, because more substrate molecules are available to bind to active sites. Non-competitive inhibition works differently. A non-competitive inhibitor does not bind to the active site. Instead, it binds to another part of the enzyme, often called an allosteric site. When it binds there, it changes the shape of the enzyme. This can alter the shape of the active site, making it harder or impossible for the substrate to bind effectively. Because a non-competitive inhibitor does not compete for the active site, adding more substrate usually does not overcome the inhibition. The problem is not simply that the active site is occupied. The problem is that the enzyme's shape has been changed. In exam language, a non-competitive inhibitor binds away from the active site, changes the enzyme's tertiary structure, alters the active site, and reduces enzyme activity. That comparison is worth remembering. Competitive inhibitors bind to the active site and compete with the substrate. Non-competitive inhibitors bind away from the active site and change the enzyme's shape. Competitive inhibition may be reduced by increasing substrate concentration. Non-competitive inhibition usually cannot be overcome simply by adding more substrate. Some enzymes also require helper substances. A cofactor is a non-protein substance that is needed for an enzyme to function properly. Cofactors may be inorganic ions, such as magnesium, zinc, or iron ions. These ions can help the enzyme maintain its shape, assist substrate binding, or participate in the reaction. A coenzyme is an organic cofactor. Many coenzymes are derived from vitamins. Coenzymes often help by carrying electrons, hydrogen ions, or chemical groups between reactions. In cellular respiration and photosynthesis, molecules such as NAD, FAD, and NADP are important because they help transfer electrons and hydrogen during energy transformations. You do not need to treat cofactors and coenzymes as mysterious. They are helpers. Some enzymes cannot work properly without them. If the cofactor or coenzyme is missing, the enzyme may be inactive or less effective, even if the enzyme itself is present. Now we can bring these ideas together into biochemical pathways. A biochemical pathway is a sequence of enzyme-controlled reactions. The product of one reaction becomes the substrate for the next reaction. In a pathway, enzymes do not act alone. They work in a chain, like quiet workers passing molecules from one station to the next. Photosynthesis is a pathway. Cellular respiration is a pathway. Protein synthesis involves coordinated steps. DNA replication involves coordinated enzyme action. Cells rely on pathways because complex chemical changes are usually not completed in one step. They happen through controlled sequences. Regulation of biochemical pathways is essential. If a pathway runs too quickly, resources may be wasted. If it runs too slowly, the cell may not make enough of an essential product. Cells regulate pathways by controlling enzyme activity, enzyme production, substrate availability, and inhibitor binding. One common form of regulation is feedback inhibition. In feedback inhibition, the end product of a biochemical pathway inhibits an enzyme earlier in the pathway. This helps prevent the cell from making too much of the product. When enough product has been made, the product acts like a signal to slow the pathway down. Imagine a pathway where molecule A is converted to molecule B, then C, then D. Each step is controlled by a different enzyme. If too much D accumulates, D may bind to an enzyme near the beginning of the pathway and reduce its activity. The pathway slows down. When D levels drop, inhibition may decrease and the pathway can speed up again. This is efficient. The cell does not need to wait until resources are completely wasted. It can respond to the concentration of the final product. Feedback inhibition is one way cells maintain balance, conserve energy, and regulate metabolism. In exams, pathway questions often ask you to explain what happens if an enzyme is inhibited, missing, or denatured. The answer is usually not just that the reaction stops. You need to think along the pathway. If an enzyme is inhibited, its substrate may accumulate, its product may decrease, and later products in the pathway may also decrease. A change at one step can affect every step that follows. For example, if enzyme 2 in a pathway is inhibited, the molecule before enzyme 2 may build up. The molecule after enzyme 2 may be produced in lower amounts. If that later molecule is needed for another process, the effects can spread further through the cell. Pathways are connected systems. This is why enzymes are so important in biology. They are not just small facts to memorize, they are control points. They determine whether reactions happen, how quickly they happen, and how pathways respond to changing conditions. Let us now practice the core comparison slowly. A catalyst increases the rate of a chemical reaction without being consumed. An enzyme is a biological catalyst, usually a protein. A substrate binds to the enzyme's active site, forming an enzyme-substrate complex. The enzyme lowers activation energy, allowing the reaction to occur faster. The product is released, and the enzyme can be reused. If temperature rises toward the optimum, Reaction rate may increase because enzyme and substrate molecules collide more often. If temperature rises beyond the optimum, the enzyme may denature, the active site may change shape, and the reaction rate may decrease. If pH moves away from the optimum, the enzyme's shape and active site may also be affected. A competitive inhibitor binds to the active site and competes with the substrate. A non-competitive inhibitor binds away from the active site and changes the shape of the enzyme. Cofactors and coenzymes help some enzymes function properly. Biochemical pathways are sequences of enzyme-controlled reactions, and feedback inhibition helps regulate these pathways. As you prepare for VCE biology questions, keep linking structure and function. The active site has a specific shape, so only particular substrates bind. Denaturation changes structure, so function changes. Inhibition affects binding or shape, so reaction rate changes. Pathway regulation changes enzyme activity, so the amount of product changes. Also, remember to use precise language. Instead of saying the enzyme dies, say the enzyme denatures. Instead of saying the enzyme gets used up, say the enzyme is not consumed and can be reused. Instead of saying the inhibitor blocks the reaction, explain where the inhibitor binds and how that affects enzyme-substrate complex formation. This kind of careful language is what turns a basic biology answer into a strong biology answer. You are not just naming terms, you are showing the relationship between molecules, reactions, and cellular control. And now, let us close the pathway for tonight. A cell is not a bag of random chemicals. It is a regulated, organized, living system. Enzymes give that system control. They lower activation energy. They hold substrates in the right position. They speed reactions without being consumed. They respond to temperature, pH, inhibitors, and helper molecules. They link together in pathways that allow cells to transform energy and maintain life. So as you rest, let the ideas settle gently. Active site, substrate, enzyme-substrate complex, product, activation energy, denaturation, competitive inhibition, non-competitive inhibition, cofactor, coenzyme, biochemical pathway, feedback inhibition. Each term is a small part of the larger story of life at the cellular level. Quiet reactions, carefully controlled, happening again and again inside living cells. And for now, that is enough. Let the pathway slow, let the mind soften, sleep well, and I will see you in the next episode.